Hello everyone, this is Akhil Chaudhary with my teammates Dheeraj Singh and Tanmay Kansal under the supervision of Professor Sachin Jain. We are working on gene prediction using deep learning and our project ID is 30. So let's dive right into it. Deciphering the genetic regulatory code that determines gene prediction is the key in molecular biology, and deep learning algorithms have revolutionized these prediction in a number of fields, and they have the potential to do it again in molecular biology and genetics. CNNs, that is convolutional neural networks, and other deep learning and machine learning technologies are largely to blame for this recent shift in image and natural language processing. But the same algorithms can be used for processing the gene data. Genes identify the characteristics of each and every living organisms, and you can say that gene is the code of life. And in order to study these genes, we can easily use deep learning algorithms like convolutional neural networks, which is currently used for facial recognition and speech recognition, or even self-driving vehicles. Deep learning methods are currently in use in molecular biology, agriculture, genetics, and medicine.
So, in the literature section, we have given the findings that we got from the previous researchers and their papers, the problems that were being faced by them in the past, and what we can learn from them. We also got to know the previous generation project methods and the extent to which they were correct and give the results accurately.
Our problem statement is to use CNN to predict gene in metagenomic fragments and to investigate the effects of CNN on gene prediction. So our proposed solution, we take a single-stranded DNA sequence as the double-stranded DNA sequence have ATGC pairs which have same corresponding base pairs in second strand of the DNA. This single-stranded DNA sequence will be used as the training data for our CNN model to find the patterns in them. The data which we will work on is similar to images, and deep learning CNN is the best architecture to use here in order to find patterns in genetic codes. Data initialization, training, data classification, and data post-processing are three primary aspects of our suggested method. Before feeding the ORF into the CNN model, we numerically encode them, and for classification step, 10 CNN models are created. Finally, the candidate ORF gene probabilities are approximated using CNN classifiers, and the final gene set is chosen using a greedy approach.
So here we have our project, and this is the basic structure of it. So here in this folder models, we have our trained model, and the CNN MGP. This is the Python source file, which will be triggering all the methods, and frag.fasta files. This is the input file that we'll be giving into our model. It contains the code for a single strand, which comprises of bases from DNA bases, which is G, T, C, and A. Uh, these are patterns for a single strand, and let's start the model. So here we are asked for the input file, that is frag.fasta, and now it's asking for a file name for the results. So let's give it name res, and now it's asking for an ORF length for the minimum ORF length. So let's give it 45. Now it's asking for the condos for which the ORS should be determined. Let's give the alternate condos ATG and TTG, CTG, and GTG. Now it's asking for the result file, what we are going to determine. Like if we want to translate it protein ORF file or a nucleotide CDS file. So let's go with the protein one. The code for it is PROT. And now our model is scanning the input single strand DNA code, and the code is completed. Let's see the result. So here we have the coords file for the coordinates of each and every ORFs, and here we have the prot.fasta file. It gives us the ORFs for each and every protein protein ORS that it is found.
Now the result. So after successfully running the project, the final accuracy report that was generated is given below, according to which we can see that at different number of filters and CNN layers, the code gives progressively increasing accuracy at the increasing number of filters, and at 200 filters and one CNN layer, we got maximum accuracy of 97.92%. Now for the conclusion part. Finally, the conclusion that we can get from this project of gene prediction is that it is a developing topic in the field of machine learning, and there is a lot of scope of improvement, as this will be helpful in the protein coding, as this can play a large role in bioinformatics.
Thank you for your attention.